Myself, Dr. Roma Khai, Associate Professor, Kite School of Pharmacy from Kite Group of Institutions, would like to tell you about the topic active surveillance. This lecture is lecture number 22 in your subject Pharmacovigilance PP 805 ET final year. So, let us begin. What is active pharmacovigilance? Now, active pharmacovigilance means that measures are taken proactively. The name itself says the measures are taken proactively. What measures are taken? To detect the safety concerns. Safety concerns means any type of harmful effects, any type of areas which are coming due to the usage of drugs, that is the medicines as well as vaccines. This is achieved by active monitoring at the start, during and at the time after treatment. So, that means at the monitoring of the drug when the patient has started taking the drug, during the treatment and after the treatment also. It seeks to find out completely number of adverse events via continuous pre-organized process. It is in contrast to passive surveillance. In my last video, I discussed about the passive surveillance. Here, I will be focusing on active pharmacovigilance, that is active surveillance. Events may be detected by screening the patient records or by direct questioning of the patients or through lab testing at predefined intervals, at different intervals. Patients who fill a prescription for the drug may be asked to complete a brief survey form and give permission for later contact. ठीक है, सपोज मैं मेरी कंपनी एक्टिव फार्माकोविजिलेंस करना चाहती है तो मैं एक सर्वे फॉर्म दे दूंगी पेशेंट्स को और सपोज मुझे कोई पर्टिकुलर ड्रग का डाटा चाहिए तो मैं उन पेशेंट्स का नंबर ले लूँगी और उनको कॉन्टैक्ट करती जाऊँगी टाइम टू टाइम, तो मैं डाटा एक्टिवली कलेक्ट कर रही हूँ. Advantages of active pharmacovigilance is it will give me comprehensive data. It helps to, it's more feasible to get comprehensive data, means detailed data on a particular individual adverse event reports through an active surveillance system than through a passive. An example is follow up of patients treated with a particular drug, like in cohort event monitoring, which I will be telling you after this slide. What are the advantages? So, advantage of active pharmacovigilance, as I have told you, that you are getting the comprehensive data, detailed data. So, active surveillance pharmacovigilance systems, they estimate the burden of adverse events. They can tell us how many adverse events are there actually with a drug, and they can generate useful information on risk factors of adverse events for more effective medicine use. Now, let us understand the methods of active surveillance. First one is cohort event monitoring. Next, we will be discussing on registries. Next, we will see what are Sentinel Sites. Then we will see the drug event monitoring. So, let us start the first one, cohort event monitoring, which is also called medicine event monitoring. As you can see, there is a prescription. तो medicine event मतलब जब एक prescription आती है patient के पास, तो तब से ये monitoring start हो जाती है. Just say it's same as nothing but prescription event monitoring. इसका दूसरा नाम क्या है? Prescription event monitoring. Studies using this process are cohort based. That means we have chosen a group जिसको हम monitor कर रहे हैं, जिनको एक medicine दी गई है और उनको monitoring की जा रही है. So, studies are cohort based. The name is cohort here, and it is prospective. Prospective means follow up, and it is observational. मतलब person is only observing. जो रिपोर्टर है वो सिर्फ ऑब्जर्व कर रहा है जो डॉक्टर ने मेडिसिन दिया है. A follow-up questionnaire can then be sent to every either physician or to a patient at some intervals, pre-specified intervals to acquire the data. हमारा मेन एम क्या है? डाटा कलेक्ट करना है. किसके बारे में डाटा कलेक्ट करना है? Regarding some safety concerns, regarding some bad effects of the drugs. So, request for data on patient demographics. और ज़रूरी नहीं है क्वेश्चन है सिर्फ हम बेसिकली एडवर्स इवेंट्स का इकट्ठा करते हैं. हम लोग बेसिकली पेशेंट का भी इकट्ठा करते हैं कि पेशेंट की हिस्ट्री क्या है और उसने जब ड्रग लिया तो उसके साथ क्या हुआ, ये प्रॉब्लम आई या नहीं आई. ठीक है. सो वी आर ट्राइंग टू एक्वायर द आउटकम डाटा. सो रिक्वेस्ट फॉर डाटा ऑन पेशेंट डेमोग्राफिक्स एज क्या है, जेंडर क्या है, इंडिकेशन फॉर ट्रीटमेंट, क्यों ये ट्रीटमेंट दिया गया, क्या ड्यूरेशन रही थेरेपी की, डोसेज, क्लिनिकल इवेंट्स क्या हुए, रीज़न फॉर टर्मिनेशन, जब आपने दवाई अगर स्टॉप करी है तो क्यों स्टॉप करी है, एंड पास हिस्ट्री. ऑल दैट कैन बी इन्वॉल्व इन अ क्वेश्चनेयर. नाउ टू गैदर मोर इन्फॉर्मेशन सेफ्टी प्रोफाइल ऑफ अ न्यू केमिकल moiety, this cohort event monitoring is done. So, it is a prospective prospective मतलब इसका follow up किया जा रहा है with time. Longitudinal, this is called longitudinal. Observational, observe करके किया जा रहा है. Cohort study एक particular group में किया जा रहा है, एक particular group को monitor किया जा रहा है, maybe children, maybe elderly, maybe a particular age group. Okay, so prospective longitudinal observation cohort study of adverse events associated with one or more monitored medicines. So what is the technique in this? Patients are enrolled into cohort, then they are followed up during treatment to record all adverse events. So it's a form of active pharmacovigilance. It's a real time monitoring over a period of time. जैसे आप देख लीजिए, एक population में से हमने एक cohort निकाला, maybe 60 years older people, ठीक है, और उनको स्टडी उसको एक्सपोज किया एक पर्टिकुलर ड्रग, हमने नहीं किया, डॉक्टर ने दिया. लेट्स टेक डॉक्टर ने कोई पर्टिकुलर मेडिसिन दी उसको, सम मेडिसिन इज गिवन, एंड देन वी ट्राई टू अंडरस्टैंड कि इस मेडिसिन से कोई प्रॉब्लम तो नहीं आई, कोई एडवर्स इवेंट तो नहीं आया. कितने लोगों में आया, कितने लोगों में नहीं आया, उससे हमें पता चल जाएगा क्या इंसिडेंस है एक पर्टिकुलर एडवर्स इवेंट का. Now basically it's a very active method, PV method promoted by WHO. It's a UK based concept. I've shown a photograph of a person called Professor Bill Inman who has introduced this particular cohort event monitoring system. जिसे introduce किया because he found there were limitations spontaneous monitoring. Doctors कई बार report करते हैं, नहीं करते spontaneous reporting system में, right? So he established a particular say research unit, drug safety research unit in UK. Okay, at the University of Southampton, and devised this method for early detection of potential drug hazards, and this method was called prescription event monitoring. So it's a method where information is collected with focus on events on all the patients being treated with a medicine or a group of medicines. Let's try to understand with the help of flow chart. This is the source of this particular chart. आप देखिए, सबसे पहले पेशेंट कहाँ जाता है, डॉक्टर के पास जाता है, जनरल प्रैक्टिशनर के पास जाता है. जनरल प्रैक्टिशनर से इसको क्या मिलती है, प्रिस्क्रिप्शन फॉर न्यू ड्रग टू पेशेंट बाय द जनरल प्रैक्टिशनर, राइट. द पेशेंट गोज टू द फार्मेसी, आफ्टरवर्ड्स मेडिसिन का उसको प्रिस्क्रिप्शन में मिल गई है, देन ही गोज टू फार्मेसी टू कलेक्ट द ड्रग. दिस फार्मेसी सेंड प्रिस्क्रिप्शन टू PPA. व्हाट इज दिस PPA? प्रिस्क्रिप्शन प्राइसिंग अथॉरिटी, राइट. प्रिस्क्रिप्शन प्राइसिंग अथॉरिटी के पास ये डाटा चला गया है. Electronic copies of prescriptions are sent to DSRU, that is Drug Safety Research Unit. So ultimately prescriptions क्या आ गई है, DSRU में. DSRU issue करता है green form, और ये green form आप देख सकते हैं, green form for details of patient events. तो ये green form जाता है जनरल प्रैक्टिशनर के पास. General practitioner fills the form. This green form question is sent back. It is sent back to the DSRU. This is the outcome data. और इस particular क्वेश्चनियर को स्टडी करता है DSRU एंड ट्राइज टू फाइंड आउट कि ड्रग की वजह से कोई एडवर्स रिएक्शंस तो नहीं हो गए. सो इट हेल्प्स इन सिग्नल जनरेशन. This data will be used for hypothesis testing. This data will be used for follow-up studies. So first of all, collection of exposure data begins soon after the new drug is launched. Then for every patient, DSRU prepares a record in the data sheet of the drug use. After prescription to the patient, DSRU sends the prescriber green form. This green form is reviewed by the medical scientific officer. They will monitor what drug was taken by the patient, what type of serious ADRs or what type of events were experienced by the patient. So it's a method of studying safety of a specially new medication used by general practitioner. तो आप देखो proactive approach है पूरा. So here the patient is being prescribed a monitored drug whose monitoring is being done. The information first 5,000 to 10,000 prescriptions is obtained. Prescribers are contacted with a questionnaire to find out what type of clinical outcomes occurred with the drug. So there was a pre-treatment questionnaire and there is a post-treatment questionnaire which have to be filled by the doctor. And then experiences are examined and the incidence of various adverse events can be estimated. तो इस तरीके से ADRs को हम detect कर सकते हैं through cohort event system. So this is done for new chemical entity, new class of medicines, very new medicines, sorry this is by mistake, medicine related to class of medicine that has previously caused problem. Cohort event monitoring is done for potentially significant adverse event observed during pre or post marketing surveillance. So this need not be there. So this is a time limited program to complement other PV activities and it is not intended to replace spontaneous reporting. वो अपनी जगह पे रहेगा और ये अपनी जगह पे रहेगा. Now, it helps in identifying unquantified risks. For example, lot of cohort event monitoring is done by New Zealand, and the program name was New Zealand Intensive Medicine Monitoring Program in 1977. Right? Drug Safety Research Unit Southampton, as I have told you, done by Bill Inman. Japan Prescription Event Monitoring in 1999. And cohort event monitoring of anti-malarial drugs is done under Pharmacovigilance Program of India. Please remember, this is more labor intensive, more costly as compared to spontaneous reporting and target reporting that was passive pharmacovigilance methods. More features, cohort is based on first prescription in the pharmacy. जिस पर्टिकुलर फार्मेसी में बेसिकली जो ड्रग दिया गया है वो किस पर्टिकुलर ग्रुप को दिया गया है वो कोहॉर्ट होगा. सो पेशेंट एक्ट एज अ सोर्स ऑफ इंफॉर्मेशन. Web based questionnaires are given, multiple questions are given. Questionnaires are regarding the patient characteristics, how much drug is used, what are adverse drug reactions, the person has got after the drug use, what is the quality of life after the drug use. So, follow up questionnaires are sent periodically to the prescribing doctors, requesting information on all the events that have occurred with the patients. So, you can see example, this was a article safety profile of artemethyl lumefantrine, a cohort event monitoring study in public health facilities in Tanzania. So, what was the methodology taken? The patients who presented to public health facilities, 4 regions of Tanzania were selected, they were prescribed AL. What is AL? Artemethyl lumefantrine. They were enrolled in the study, and then it was found that how many adverse events were reported. So, it helped to characterize the known reaction, it detects signals of unrecognized reactions, it identifies interaction with other medicines, क्योंकि एक cohort को आप observe कर रहे हो continuously. It detects inefficacy of medicine, it assesses safety in pregnancy, lactation, identify risk factors. So, objectives are to characterize known reactions, कि कौन सी particular age में ADR हुआ, क्या जेंडर था, क्या डोज दिया गया, क्या ट्रीटमेंट ड्यूरेशन थी, क्या टाइम टू ऑनसेट था, क्या सीरियसनेस प्रोफाइल थी, कितने लोगों में इंसिडेंस हुआ, क्या आउटकम था ड्रग का, वेदर पेशेंट्स विड्रॉन द ड्रग, सो व्हाट इज कोई सिंड्रोम न्यू आ गया उस ड्रग की वजह से. Detect signals of unrecognized reactions, it can tell you the interaction with other medicines, it can tell you the interaction with complementary alternative medicine, it can tell you food drug interactions, it can help in identifying the risk factors so that they can be avoided. What was the age in which it was given, gender, dose, duration, what was the person in whom the drug was given, जिनमें दिया गया था उनमें कौन सी other diseases थी, उनमें कौन से और other medicines वो ले रहे थे. तो ये सारे रिस्क फैक्टर्स को ये स्टडी करता है कोहॉर्ट इवेंट मॉनिटरिंग. राइट. Assess safety in pregnancy and lactation, it estimates risk, it provides evidence for effective risk management, safer prescribing, benefit assessment, regulatory changes. तो हमें ये एविडेंस मिलेगा तभी तो हम ये बेसिकली काम कर पाएंगे. Sorry. It detects inefficacy which might be due to faulty administration, poor storage condition, out of date, poor quality product, counterfeit medicines and interactions. तो ये सारे objectives हैं. तो ultimate objective क्या है? Achieve maximum benefit from a medicine and least harm from a medicine for our public, for our patients. Next method of this active pharmacovigilance is registry. What is a registry? एक register होता है. register में इस तरह से हम attendance register बनाते हैं. इस तरह का register में क्या होता है? list of patients presenting with some characteristics. So, there can be disease registry, there can be drug registry, there can be pregnancy registry. तो ये registries जो जिनको हम patient registries भी बोलते हैं, these are systems that use observational methods to collect uniform data on a population, either defined by disease, condition or exposure, and then they are followed over time. Disease registry में क्या होता है? Composed of the number of patients, list of patients who have had a disease or a particular condition of interest, जिसको हम पढ़ना चाहते हैं. Product registry, they are composed of patients exposed to a healthcare product, so they allow the study of what is the drug utilization patterns. Pregnancy exposure registries focus on exposures during pregnancy, postpartum is after delivery, also affects in the offspring. तो pregnancy exposure registry में वो डाटा आता है कि कौन से ड्रग्स प्रेग्नेंट वुमन में दिया गया, आफ्टर डिलीवरी दिया गया और उसके क्या इफेक्ट्स आए बेबीज पर. सो registry, as I told you, it's a list presenting with the same character. This characteristic can be a disease registry. Let's see the examples, or a drug registry. Both type of registry differ by the type of patient data of interest. They are collecting information using standardized questionnaires. Let's see example of disease registry. Disease registry example, registry for blood dyscrasias. Simple word, dyscrasias क्या होता है? Disorders. कितने लोगों में blood disorders problem आई? Registries for severe cutaneous reactions. Registries for congenital malformations. तो ये disease registry हमें डाटा देता है. It can help collect data on drug exposure, which drug was given, and which other risk factors which have caused these diseases. Blood कौन से drug थे जिसकी वजह से blood disorders हुए. So they can also act as a base for case control study. अगर हमें पता चल गया कि एक particular drug ये disease कर सकता है, तो ये सारा हम एक hypothesis बना सकते हैं और उसको हम base बना सकते हैं for a case control study. And then we can, what we do in case control study, we compare the drug exposure cases identified from the registry and the control selected from either patients, okay, with some other condition. Okay. Drug registries, they address populations exposed to drugs of interest. For example, registry of rheumatoid arthritis patients exposed to biological therapy. तो यहाँ drug क्या है? यहाँ drug है आपका biological therapy, right? This helps to determine if a drug has a special impact on this group of patients. कौन सा group है? Rheumatoid patients. Some drug registry address drug exposure in specific populations also, like pregnant women. So patients can be followed over time and included in cohort study to collect data, further data on the adverse events which has happened by taking the medicine. And this data is collected using questionnaire. It is also useful for signal amplification. को signal मिल गया है, signal amplify करना है, मतलब इसका मतलब और data हमें add up करना है. तो this particular drug registry help us, especially for rare outcomes. It can be very valuable when examining the safety of orphan drugs. Orphan drugs, please remember, they are given for rare diseases. Okay. Next type of active surveillance is Sentinel Site. Now, what is Sentinel Site? Sentinel Site एक site है, जो एक community है, जहाँ से हम काफ़ी ज़्यादा data collect कर सकते हैं, from which in-depth data can be gathered and analysis can be conducted. It is used to deal with the programs and policies affecting a large geographical area. Sentinel Site generate results that are representative of broader population. यानी कि ऐसे results है जिसको हम कह सकते हैं कि सारी population में ये problem आती होगी. It can be मतलब it can be interpreted for broader population. The concentration of resources in defined geographical areas produce a rich source of information that would have been very costly if implemented on national scale. तो हम एक basically हम ऐसी communities choose कर लेते हैं basically जो वहाँ जो drug use किया जा रहा है और उस पर हम डाटा कलेक्ट करते हैं उस पर. Active surveillance can be achieved by either by reviewing the medical records, maybe in a hospital, maybe in a clinic, or by interviewing patients, or by interviewing physicians in a sample of Sentinel Site to ensure complete and accurate data is received on the reported adverse events. The selected sites can provide information like we can get data from specific patient subgroups that would not be available in a passive spontaneous reporting system. Further information, the use of drug, maybe हम drug abuse centers में जा सकते हैं और वहाँ पर जो लोग use मतलब ऐसी क्या drugs use हुए हैं जिसका abuse हुआ है तो हम information collect कर सकते हैं. But the major weaknesses, problems with selection bias. Selection biases may be जो हमने select कर रहे हैं centers वो ठीक नहीं है. Maybe there is some we should not have selected those centers. Small number of patients are there, increased costs, so that is the drawback. It is most efficient for those medicines which are used in institution settings such as hospitals, nursing homes, hemodialysis centers. So students, these settings can have a greater frequency of use for certain drug products. जैसे de-addiction centers होते हैं तो वहाँ पर एक particular जैसे निकोटीन गम आया तो smokers को दिया जाता है, right? So institution settings can have a greater frequency of use for certain drug products. Automatic detection of abnormal lab values from computerized lab reports in certain clinical settings can provide the efficient active surveillance system. We can get detection of abnormal lab values from such patients. Intensive monitoring of sentinel sites can helpful in identifying risks among patients taking the orphan drugs. Let me show you, this is a COVID center, okay, COVID care center, so that becomes a sentinel site. So, let us have a summary. Active surveillance carried out in institutions, nursing homes, clinics, hospitals, COVID care centers. So, they form a sentinel site. So, वहाँ से हमें क्या मिल सकती है? Information through questionnaire. So, they provide information such as data from specific patient subgroups. तो हमें different subgroups की information मिल सकती है, अलग अलग age groups की मिल सकती है. By questioning patients, by questioning physicians, and this will guarantee that comprehensive and precise data we can collect from these sites. The last type of this active pharmacovigilance or active surveillance is EHR, electronic health record mining. Electronic health or medical records are rich source of ADR data. We should know that health records, they have a rich source of ADR data. ADR, again I am telling you, it's adverse drug reaction data. They contain detailed patient information and abundant longitudinal data. Longitudinal data जब भी मैं बोलती हूँ उसका मतलब follow up किया जा रहा है time intervals पे patient का. EHR mining can either complement and improve ADR monitoring program. अगर हम पर्टिकुलर रिकॉर्ड्स में जाएँ, सेम पेशेंट का रिकॉर्ड देखें, एक पर्टिकुलर ड्रग वो यूज़ कर रहा है, एक से ज़्यादा ड्रग यूज़ कर रहा है और उसको टाइम टू टाइम हम चेक करते जाएँ उस डाटा को, तो हम फाइंड आउट कर सकते हैं कि क्या कोई पर्टिकुलर बैड इफेक्ट ही हैज़ गॉट इट, ही हैज़ एनकाउंटर्ड इट. सो दैट वी विल कम टू नो दैट कैन बी बिकॉज ऑफ सम ड्रग. So, here the denominator is known. We know the total number of people exposed to a drug is known. It addresses all the events, it covers all the medicines, and done by using data available in patient records to enhance the pharmacovigilance. So, basically, so this is the last part, electronic health mining. This was the last part of the active surveillance. So, let me just do the recap. Okay, just if I do the recap. So, first of all, in active surveillance we have seen that active measures are taken to collect the information. The first one was cohort event monitoring, which is also called prescription event monitoring. It was started by Bill Inman. Then we saw registries. They can be disease registries, they can be drug registries, they can be pregnancy registries, right, based upon the exposures. Third one, if we see, we have seen sentinel sites. That means, they are special community sites from which the data is collected, right. And last one was mining record. If we simply take the data from the records, medical records, and we can follow up the patients and all, so we are able to find out the ADRs. We can address all events. So, these are the references from which this data has been picked up and I have compiled. So, you all are requested to go through all these textbooks, and the slide share also you can go through this, and thank you all for patient listening. I hope you have understood what is active surveillance. In the last video I told about the passive. So, you can see the comparison between passive and active. In passive there were no active measures were taken, and here there are proactive measures taken for the monitoring of the drug. Thank you all.